The year was 1935. Farmers in Australia had a problem. Sugarcane fields were under attack by beetles, threatening an entire industry. Scientists had a solution: introduce cane toads, a species from South America, to devour the pests. It seemed logical, calculated, controlled. But within years, the toads multiplied uncontrollably, spreading across the continent. They didn't just eat the beetles. They poisoned predators, devastated ecosystems, and created a crisis far greater than the one they were meant to solve. A simple intervention spiraled into an unstoppable force. For centuries, humanity has sought to control nature, bending it to our will. We dam rivers, alter landscapes, and now, with bioengineering, we are rewriting the very code of life. The logic is the same: identify a problem, introduce a solution. But nature has never been a passive player. It responds, adapts, and often in ways we fail to predict. Today, we wield tools far more powerful than introducing a single species. With CRISPR and gene drives, we can edit DNA, modify entire populations, and even attempt to eradicate diseases. Imagine mosquitoes that can no longer carry malaria, crops resistant to pests, supercharged bacteria designed to break down plastic waste. The potential is extraordinary, but so is the risk. History has shown us that when we intervene in nature, unintended consequences follow. The more precise our tools become, the more unpredictable the response. What if a modified organism evolves beyond our control? What if a self-replicating gene spreads through ecosystems in ways we never anticipated? If releasing a single species of toad led to environmental havoc? What happens when we begin altering life at its most fundamental level? Perhaps the real question isn't whether we can control nature, but whether nature will allow itself to be controlled. In the 1940s, farmers faced a new challenge: weeds choking their crops, stealing nutrients, and reducing yields. The solution seemed revolutionary: herbicide-resistant plants, genetically engineered to survive powerful chemicals. At first, it worked. Crops flourished while weeds withered. But nature didn't surrender. It adapted. Within a few decades, superweeds emerged stronger, harder to kill, evolving defenses against the very chemicals designed to eradicate them. The battle escalated, forcing farmers to use more herbicides, creating an arms race between human innovation and nature's relentless adaptability. This wasn't an isolated case. Antibiotics, hailed as one of the greatest medical breakthroughs, followed a similar trajectory. The more we used them, the faster bacteria evolved resistance. Superbugs emerged, strains of disease causing bacteria immune to the drugs meant to destroy them. In hospitals around the world, once treatable infections now defy medicine, spreading unchecked. Each solution led to a new, unforeseen problem. Now we stand at the frontier of genetic engineering. CRISPR technology allows us to edit DNA with precision once thought impossible. Scientists are deploying gene drives, genetic modifications designed to spread through entire populations, permanently altering species. Malaria-carrying mosquitoes are being programmed to pass on genes that prevent them from reproducing. The goal? Wipe them out completely. But what if nature fights back again? What if the mosquitoes evolve resistance? Or worse, a mutation emerges? That makes them even more efficient carriers? We are engineering solutions at a scale never seen before, yet the unintended consequences remain unknown. Could we be triggering a new kind of evolutionary arms race? One we can't control? If small genetic changes in weeds and bacteria led to unstoppable adaptations, what happens when we start rewriting the genetic code of entire ecosystems? In 2018, scientists in a laboratory altered the genes of mosquitoes, engineering them to carry a self-destructive trait. The idea was simple: release them into the wild, let them breed, and within, some of the mosquitoes survived. Not only did they evade extinction, but they carried genetic fragments of the engineered strain, mutations that scientists hadn't predicted. These altered mosquitoes spread, stronger and more resilient than before. Instead of being eliminated, they adapted, evolving in ways no one had foreseen. The very technology designed to eradicate them had, in some cases, made them harder to control. This isn't an isolated concern. Across the world, gene drives are being explored as a way to reshape entire ecosystems. Scientists are working to engineer coral reefs that can withstand rising temperatures, trees that absorb more carbon, and even crops that produce their own pesticides. On paper, these solutions seem promising, but nature is not a machine with predictable outputs. It is a web complex, interconnected, and constantly shifting. A single genetic change in one species could trigger cascading effects across an entire ecosystem. Imagine modifying a plant to resist pests, only to discover that pollinators can no longer recognize its flowers, or engineering a fish to grow faster, only to watch it outcompete and drive other species to extinction. When we tinker with nature's balance, we don't just change one thing, we change everything connected to it. We are now rewriting the blueprints of life itself. But if nature has always found a way to push back against control, how long before it does so again? And what happens when we're no longer able to predict the outcome? In 2020, a team of researchers in China created genetically modified rice, designed to resist drought and pests. The experiment was hailed as a breakthrough, a way to feed a growing population in an unpredictable climate. But then, something strange occurred. Nearby wild rice, untouched by human hands, began to change. The modified genes had spread, blending with natural varieties in ways scientists hadn't anticipated. The result? A strain of rice that was harder to control, more aggressive, and resistant to traditional farming methods. The boundary between engineered and wild had blurred. This phenomenon, known as genetic spillover, is not just a theoretical concern. When modified organisms interact with natural ecosystems, the consequences can ripple outward in unexpected ways. In Canada, genetically modified canola plants, designed to be herbicide-resistant, escaped into the wild. They formed new hybrid species, spreading across roadsides, fields, and forests, altering local biodiversity in ways that scientists had not predicted. Now, as we push deeper into bioengineering, the risks only grow. In labs across the world, researchers are designing synthetic bacteria to clean up oil spills, break down plastics, even generate electricity. These microorganisms are programmed with safety features, genetic kill switches, meant to prevent them from surviving outside controlled environments. But nature doesn't follow human rules. What if one of these organisms mutates, finds a way to bypass its programmed limits, and spreads beyond its intended purpose? It's not just a question of science, it's a question of responsibility. How do we test for every possible consequence when the interactions are limitless? If a single modification can escape into the wild and rewrite the balance of an ecosystem, how do we ensure we're not setting off a chain reaction we can't reverse? In our pursuit of control, are we creating forces that may soon be beyond it? For centuries, humanity has shaped the world to its needs, cutting through mountains, rerouting rivers, and now rewriting the very fabric of life. But nature has never been a passive canvas. It responds, shifts, and adapts in ways we don't always expect. The more we try to control it, the more unpredictable it becomes. Consider the Yellowstone wolves. In the 1920s, they were hunted to extinction, seen as a threat to livestock and game. Without them, deer populations exploded, stripping vegetation and destabilizing the entire ecosystem. Decades later, wolves were reintroduced, and almost immediately, nature corrected itself. Rivers changed course, forests recovered, and balance was restored. Now we are making far greater changes, editing genes, engineering species, and creating organisms that have never existed before. But unlike the Yellowstone wolves, these creations don't have a natural blueprint to follow. Once released, there is no way to call them back. No reset button, no second chance. Perhaps the question is not whether we should use these technologies, but how we use them. Can we proceed with caution, ensuring that each step is measured, each consequence considered? Or will our attempts to perfect nature push it towards something unrecognizable? The future of bioengineering is not just about what we can create, it's about what we might unleash. And as we stand on the edge of this new frontier, one question remains. When nature strikes back, will we be ready? This video has come to an end, but the discovery continues. Check out more content on the channel and subscribe so you don't miss what's next. Thank you for watching.